First, I really want to thank the dream organizers to give us this opportunity. Actually, our team really had a lot of fun in doing this project because we learned a lot during the analysis process. Also, we learned a lot when we try to interpret the results. Actually, honestly speaking, when we did analysis, a lot of decisions was made based on our intuition. So until we look at the result, we found, oh, actually the result is pretty good. Then at that time point, we try to think really very clearly what is the rationale behind those intuition, and we found actually a lot of intuition. It turns out to be correct. So now I would like to share our experience with you today about this challenge. The title of my talk is Predicting Synergistic Effect of Compound Combinations Using Genomics Data. Here is a little background about this challenge. Due to the complexity of biological processes, single drugs usually do not work very well for multifactorial diseases such as cancer. Drug combinations are one of the most promising ways to work treating these diseases. And however, some big screening experiments show that only 4 to 10% of drug pairs are synergistic. So it is important to predict which drug pairs are synergistic pairs. Genomic information has been used to predict synergistic drug combinations. So the goal of this challenge is to use genomic information to build models to rank the sensitivity of cancer cell lines to a set of 2-component combinations of small molecular compounds. So as mentioned by previous speaker, this is the definition of synergy by this challenge. Use the Bliss score. So here, C is the expected combination effect, assuming compound X and Y are independent. And then the score is the difference between the actual combination effect and the expected effect, C. So in order to get this score, we need to estimate what is the actual combination effect. So this is the goal of this challenge. So this is the data provided by the challenge. Since the previous speaker already mentioned this, so just very briefly. So for the genomics data, we have gene expression profiles after 14 perturbations at 3 time points and 2 concentrations, and also baseline profiling data, and also the genetic data. For the for the dose response data, we have the dose response curve at 24 hours and 48 hours. Please know that for this particular challenge, there's no training data. So we do not know any experimental data, the Z, for any combinations. So we cannot use any supervised approach. We have to use an unsupervised approach. So now I will talk about some basic hypotheses that we have to approach this challenge. So these are cells on plate, and this is the baseline genomic profile. So after the cells are treated by compound A, a fraction of the cells were killed by compound A. We denote as FA. For example, if we use IC20, then this FA is 20%. For the remaining cells, Y minus FA, the genomic profile has been changed. So we hypothesize that this genomic change due to the compound A will have some residual effect on the drugs, on the cells. So we denote this effect as A prime. Then we compound B. Now the question is, what is the fraction of cells will be killed by compound B conditional on this genomic change induced by compound A? So if we can calculate this value FB plus A prime, then we can solve the problem to get the combination effect Z. So now our goal is to estimate this parameter FB plus A prime. In order to do that, we define a correlation R that measures the similarity between compound A and compound B. So there are two boundary cases where we can estimate the induced cell death Z. The first case, R equal to 0, that means compound A and compound B are independent. In this case, it is very easy. So this is effect of compound A and compound B. Then the effect is we just use this formula because they are independent. So that means the genomic profile change due to compound A does not have any effect on compound B effect. Another case is R equal to 1. That means compound B has the same effect as compound A. So that means the genomic change due to compound A has the same effect as compound B. In this case, we can use this formula. So basically, the effect due to compound A is just equivalent to add the same amount of dose for compound B. So this is the formula for Z. So now we need to estimate F2B. It is easy because we have drug response curve. So using this drug response curve, so we can easily calculate this is the fraction of cell death due to drug compound B. This is just F2B. So using this curve, we can get this estimation. Next, we will just try to generalize this idea. If the similarity between is something between 0 and 1, then what we did is we just project the effect of A into B. And then use this RF2B. That means that is similarity between the compound A and compound B. And then we just use this formula to calculate the Z. Okay? So this is how we estimate the drug combination effect between compound A and compound B. So basically here, FA is the killing effect coming from A only. And this part is the killing effect come from B and A. That is the similarity part between B and A. And this is the dissimilar part of B. Now the question is that this formula is asymmetric. What does that mean? That means that if we add A first and B last, then this is the formula. But if we add compound B first and then add compound A, the formula will be the same, but probably the parameter is not symmetric. So that means it will probably lead to the different effect when we add two drugs in different orders. Based on this, then the first thing we check is whether this assumption is reasonable or not. So we check the literature. Actually, this morning, I'm very glad to hear Dr. Joe Gray mention that the sequencing or the order does matter. So actually, we look at the literature, and there's a very interesting paper published in Clinical Cancer Research said that appropriate sequencing and the scheduling of chemotherapeutic agents may overcome some drug resistance. They give an example of the combination of cisplatin and paclitaxel and shows that if add cisplatin first, actually it will reduce drug paclitaxel sensitivity. But add paclitaxel first, it will actually induce sequence-independent synergy. Similarly, for the combination of flavopiridol and paclitaxel. So they give some biological interpretation of this because that is related to the cell cycle pathways. And another paper from a big clinical trial also shows that giving chemotherapy first has better overall survival results than giving radiation therapy first. So because of this evidence, we felt comfortable to use that model. But for this particular challenging, we were not given the ordering or the scheduling of the two drugs. So what we did is that we just calculate the effect of ZBA, that is adding B first and A, and as this, and also calculate the ZAB, and then just get the average of these two. So this Z is what we estimate the overall combination effect. So because of this, we can see that based on this formula, everything we can get everything except this R. Remember, R is the similarity coefficient between compound A and compound B. So now the only thing left is how to estimate this coefficient or the similarity. So we use a very simple model. So first, we just look at the gene expression profile, and then to identify the up and down regulated genes after precipitation of compound A, then we can have two gene lists, down-regulated A and up-regulated A. And similarly, we can get the down-regulated gene due to the compound B precipitation and up-regulated gene list due to the compound B precipitation. And then we just compare these four gene lists. If one gene belongs to both down-regulated due to compound A and down-regulated due to compound B, then the similarity score will increase. Similarly, if there's overlap between up-regulated due to compound A and compound B, will also increase the similarity. On the other hand, if one gene is down-regulated by compound A but up-regulated by compound B, then the similarity score will decrease. So this is the basic formula we use to estimate similarity coefficient between compound A and compound B, to just get the overlap among these four gene lists. However, we have one consideration, just actually similar to the consideration that the Fin team had, that not all genes are related to cell growth and cell viability. That means in the gene list I showed before, probably a lot of genes are not related to this process. If we include those genes in the model, probably it will introduce more noises than signal. So we made a modification. So only genes that are related to cell growth or cell viability will contribute to the score. So we hypothesize that the only genes included in this, we call that cell growth related pathways, CGP, are important. But now the question becomes, which pathways? We don't know. So in order to identify this pathway, we look at the CAC pathway, but there's many pathways in that database. So what we did is that we searched the literature and identified a training data set for synergistic effect that was published by Jin and others in bioinformatics last year. In that paper, they were trying to study the combination effect between docetaxel and gefitinib. So they used a different dose combination of these two drugs. And then they also had the gene expression profile. So we contacted the authors and got the data for this data set. And then we used the model that we proposed to try to do the prediction. And then, because there are so many different types of the pathway combination, we just select the pathways that lead to the best prediction results. So this is the result we got for that data set. So this is the correlation between the predicted combination versus the real rank. And the Pearson correlation is 0.52. So using this, we select we call that as cell growth related pathways. So this is the pathways we selected. Altogether, we select 8 CAC pathways. And we found that actually these pathways, it seems to be really relevant to the drug response, especially from Lawrence talk. You can see that, for example, here we have NF-kappa-B signaling pathway, cell cycle pathway, P53 signaling or DNA damage pathway, apoptosis, and cancer pathways. So after that, we select these 8 CAG pathways. Altogether, there are 835 genes in these 8 pathways. So use this, we refine the similarity score by overlapping the previous gene list versus these CGP. So only genes that belong to this CGP contribute to the score. So that is the first modification. So the next consideration is that in the previous version of similarity score, we did not consider the gene-gene interactions. So we think actually the interaction also is important. So in order to do that, we look at the 30 CAG global pathways. So this is just illustration. So for example, one gene belongs to this upregulated gene set A. So this is the gene belongs to upregulated gene A. And then we look at the pathway of this gene, found there are several genes regulate this gene. This gene is kind of activate this gene, and this gene inhibit the gene. And then you see, so if this gene, the activation gene, also regulates by compound B, then actually we increase the similarity score. If the gene that inhibits this gene, but it is downregulated by gene B, then we also increase the similarity score. Basically, we consider both the gene expression direction and also the inhibition or activation in the pathway. And that, after consider that, we modify our similarity score by this formula. So basically, we expand the gene list by considering the global pathway. So this is just one example for a combination of blebbistatin. This I just call as drug A and the hydrochloride, drug B. So this is a pathway, the cancer-related pathway. And then for this, we can see that for drug A upregulated VEGF, although drug B did not upregulate VEGF, but it upregulated the upstream gene, HIF alpha. So because of this, we increase similarity score. Similarly, for another gene, also actually compound A downregulated VEGF, but compound B did not, but it also downregulated the upstream gene. So we add another score, we increase the similarity score. So that is the basic rationale for us to define the similarity score R between compound A and compound B. So after this, actually we can derive the similarity score for all compound pairs. I remember after we have the similarity score, we can get the Z, that is combination effect of all pairs. And then from Z, we can easily get the synergistic score, Bliss independent score. So this is the result we predict for all the compound pairs. And then, so after that, we see whether there's any evidence to support these ranks. So we searched the literature and found one top rank number 3. We found actually there's a publication to support the effect of these two combinations has the effect on the proliferation and apoptosis in cancer cell lines. So we submit the result, and the overall C index is 0.6. So here are some conclusions from our analysis. First, genomic change induced residual effect contribute to the synergistic effect of drug combinations. Also, the sequencing of treatment matters. The dose response curve is useful because we use it to calculate F2B. And filtering genes by cell growth-related pathway helps to focus on only cell growth-related factors, including contributions from the pathway information further improve the prediction. Actually, there are many things more things we can do, because of time limitation. Actually, we only really focus on small fraction of the whole analysis. For example, we only use the gene expression to do the analysis, but how about the other genomic information like the genetic change? And also, the other pathway and the gene network information could also help the prediction. Here, we'll only use static CAC pathway. We can also use protein-protein interaction network and also the construct gene network to see whether the result will be improved. Also here, probably we can have also better way to define up and down regulated genes. And also because at that time point we did not have the training data, so now after we have the training, we know the risk score, whether we can use some supervised approaches to do analysis. So here is the acknowledgement. So our UC Southwestern Medical Center dream team, these are the team members. Actually, we really had a lot of fun to work on this project. Our team kind of had a weekly meeting during those two months, and we learned a lot from this project. Especially Ji Chengyang and Hao Tang implement most of the analysis for this project. And we really want to appreciate dream organizers, and these are the funding agents. Thank you.
Thank you. Okay, so we are a little bit late, so we will make a break before the